Um, Thanks everybody to come to a new talk, a new seminar of the IATA Institute. Today we have a special guest because Gabrielle Berg, she's one of the most interesting researchers in the field of the microbial management. So we're gonna talk about a little bit about her. She obtained her PhD in 1995 in microbiology in the Rostock University in Germany. And then she moved to Austria where she became full professor in environmental biotechnology in the Graz University of Technology. And now in the 2021, she obtained the professorship at the University of Potsdam at the Leibniz Institute of Bioeconomy. She is working in the research of host-associated microbiomes and the translation to applications. And in the last 3 years, she belonged to the most influential researchers worldwide in the top 1%. So that's the size of the person who is going to talk today. She's going to talk about microbiome management for plant and planetary health. So thanks, Gabriel, for accepting our invitation. And here, you can start.
Yeah, thanks a lot, Roberto. And it's a pleasure to present the lecture, although I would love to be there.
Ah, yeah, it would be nice.
Yeah, and we are really all missing the scientific exchange. However, so I hope that I can present some of our research also here. So, but I would like to invite you to have a view in here, my home country, that is Styria. So we have a lot of mountains. There is already a snow layer on the top. So, and I have two affiliations, as Roberto already mentioned. So my main affiliation is in Graz here at our University of Technology, and another one, this is Leibniz Institute in Potsdam, together with the university. So, and this is my group. So this was the first party this summer. So therefore, we were all happy to be together and celebrate that. Now the situation is not that good anymore. However, so our institute is full and we are still working. I forgot to mention our vision of my group is a microbiome management for a healthy planet. So for a long time, I was working on plant health, but we extended also that scope. As an introduction, I think you are all aware that we are living in the Anthropocene, and so the man-made planet, and that the major drivers as human population growth or urbanization, globalization, overconsumption, and especially intense agriculture lead to a change of our planet, which was here visualized by one of my co-workers. And scientists from the Stockholm Resilience Center and another Leibniz Institute in Potsdam, so they've formulated the planetary boundaries. And so we crossed already for several points. We crossed it for biodiversity, for climate change, for biogeochemical flows, and I think also for novel entities. And this is the plastics which is added to our environment. So what all scientists figure out that together, especially in a synergistic way, these factors lead to or change the resilience of our ecosystems. So, and less is known at microbiome scale, and this I would like to show you today. I hope that you are all aware of the microbiome definition. For those not, I would like to repeat it here also in the introduction. So we differentiate between the microbiota. So these are only the organisms, so all small microorganisms as bacteria, archaea, the prokaryotes, but also eukaryotes, fungi, protists, and algae. Together they belong to the microbiota. And what you can see here on the right-hand side, these are algae. It's blood algae, so they have a red color in nature. And you can see that they are closely connected to bacteria, to archaea, and maybe also to protists of a different shape. And altogether, they also form a kind of house or theater. So due to their excretion products and as polysaccharides in a biofilm, but also a lot of lipids, amino acids, and relic DNA is there, but also mobile genetic elements and viruses and phages. So the viruses, they belong to the microbiome, but not to the microbiota, because they cannot they cannot live, or they are not living organisms. So, and my talk today will focus on the plant microbiome, and I would like to show you that it is interlinked also with other environments, and that there is already a loss of or a depletion of the plant microbiome, which then bring us to the second, much shorter topic, the microbiome management. So the first point is the plant microbiome is interlinked, and I think this figure shows it well. So we have a seed, there's already a seed microbiome, but of course, if the seed is germinating, so then we have also an acquirement of the soil microbiota to the plant microbiome, and then in the food or in a root or whatever we are using, we are eating that, and at the end it's then also in our human gut. Also in the air, it is clearly connected and linked to human health, but to animal health as well. So, and I selected, is maybe a little bit unusual for your institute here, but this is what I have done for a long time, is to go into natural environments and study the plant microbe interaction under those conditions, because of the fact that we expect that here this interaction is really working and not disturbed. So, and when I was here in, this is Rostock, and when I started my research, I selected these bog ecosystems because I thought they are really untouched, and we can learn a lot for plant microbe interaction. But now these ecosystems, so they have much more importance, because we are aware that this is one of the most important carbon sink which we have if they are intact, and they have a very high productivity, and they are not only a carbon storage, they are also a source for fresh water and biodiversity. So, and specific thing of these bog ecosystems is that the whole vegetation and also the whole microbial life is embedded here in the mosses. Here you can see this sphagnum mosses. So they are the engineers of these bog ecosystems. And for us, it was really interesting to study then the plant microbe interaction. So here is also these ecosystems, they are ombotrophic. So that means they live from the rain. So there's no soil below because they live on the dead mosses. So and therefore they need a lot of minerals and nutrients, and some plants have to catch insects for that even. So for us it was really a surprise, because we thought, due to the fact that everything is embedded in these mosses, they share a lot of the microbiota. This is also really true, but on the other hand, all the plants inside of that ecosystem, they show an extraordinary high degree of plant specificity. And we studied these bog ecosystems in Norway, in Germany, in Austria, and Russia, and we were really surprised about this similarity. So it was completely independent of the region. And we were wondering how that can happen. And then we figured out that inside of the spores, the microbiome, a core microbiome, is transferred, and all these small red organisms here, these are bacteria, and they form a core microbiome of beneficial bacteria. So later on, they also colonize the gametophyte, and you can see here nicely that within the phylosphere, you have a lot of different colonies and microorganisms. And in this case, the microorganisms are really essential for the plant. And this I would like to show with that figure, which is a summary, a graphical summary of a recently published study from us, also about bog ecosystems. So here you can see one of these bog ecosystems in Austria. So also in the alpine environment, there are a lot of these bog ecosystems. They are not that large as in the northern hemisphere, but there are many. So, and I think this figure here showed nicely that the plant microbiome contribute to the plant functioning, and these are here these green functions. For example, they supply nutrients. So we have done here a metagenomic study and figured out all the functions which are in that associated microbiome, and so they provide a lot of nitrogen, phosphorus, even carbon. And so they promote growth of the plants by phytohormones, for example. They interact a lot, so they are really motile, and they protect the plant against biotic and abiotic stress. And this is also important in that environment, because these bog ecosystems, they have a low pH, and normally they would be permanently attacked by fungi, but it's you can rarely see any infection or any plant disease here. So this are the functions of the bog microbiome for the plant themselves, but beyond, they have also a lot of function for the entire ecosystem. This is what I already mentioned. They fix carbon and provide that to the plant. So that is not often the case, that because of photosynthesis normally is such an efficient process. But in the carbon cycle, they have another important function, because we have here the green sphagnum, and then we have the dead sphagnum, and then we have the peat. And in peat is, on one hand, it is a carbon storage, but there is also a release of methane. So, and the sphagnum associated microbiota forms also a cooling net, because they catch the methane which goes into the air. So this is a very interesting process by methane oxidation, and contributes a lot to our world climate as well. So, and then of course they have also, for the nitrogen cycles, they have important functions. So, and it's even more here in detail. I would like to tell you what we can learn from the analysis of native plant-associated microbiota. We thought it would be interesting to look also in the rhizostome. So this is part of the microbiome function. And you know that antimicrobial resistance is one of the main topics of or challenges which we have. And therefore, it is also important to understand antimicrobial resistance in native environments. And so this we have studied here also by metagenomes. So, and then we assembled metagenomes, assembled MACs and genomes and isolates. And what we figured out is that the potential for antimicrobial resistance was unbelievable high into that native ecosystem. So this was really a surprise. We found also a lot of clinically relevant antimicrobial resistance genes, but I will explain you in the next slide why maybe it's important. And this is in the summary. So, but first part of the summary. So I always like also to study the native plants and ecosystems, because they are important to understand the microbiome functioning at mechanistic level. And so, and these native ecosystems, they are important reservoirs for new discoveries as novel strains, rules, or compounds. And for example, these mosses, so they are well-defined mini ecosystems. So they always harbor a stable and similar microbiome, and that makes them to an ideal model organisms. And for the rhizostome, I would like to show you here our conclusion. So in this, based not only on the box study, so we have done other studies as well. And what we have seen is that all native environments, which are untouched and pristine, so they have a high microbial diversity. And in accordance to that, they have also a very high diversity of antimicrobial resistant genes. So they are responsible for the plasticity of the ecosystem. And in nature, they very often have completely different functions than in hospital. But they are also at very, very low abundance, because it's a burden for the microbiome to have that genes, but they are essential for survival. So what we are doing is that man-made microbial resistance, so we are reducing microbial diversity, and we are also reducing the antimicrobial resistance genes diversity, but we enhance and enrich the pressure of specific resistance genes, and altogether this forms a risky microbiome. So, and what I have shown you here, this is also a little bit the tendency which we are seeing for the plant microbiome in general. And this is, I would like to show you now here in that part, which is still part one, for 3 examples. So one is a pumpkin, tomato, and then again, I would like to end with a native environment. So we selected the pumpkin, and it is a specific cultivar. It was a mutant found here in Styria 100 years ago, and it is now called the oilseed pumpkin, and it's one of our main crops here because we produce a lot of tasty and healthy oil from it. And why have we selected that vegetable? So it was very interesting because we have the whole breeding line. So for all our crops, it's always difficult to find all examples and to have them. And what we could see in that analysis was that breeding shaped completely the composition of the microbiome, and also it shaped the composition of it shaped the function of the microbiome. And what we have seen is also a correlation of the resistance of the pumpkin and the seed microbiota. And we have done it together. It was a study with Saatzucht Gleisdorf. So they are the breeders of that pumpkin. And what they are doing now is they integrated microbiome analysis into their breeding pipelines. This shift due to breeding was reported now many times, but normally the breeding was for bigger foods, more sweet foods, or for resistance. And also these breeding directions, they are highly visible in the microbiota. For example, all these resistant varieties, so the resistance-based, sometimes, of course, on genes and on the plant genome itself. But in many cases, it was figured out now that it also is due to the associated microbiome. And this is really interesting. I mean, it was done secretly and it was not targeted, but it is obvious that there is a correlation, which was published, for example, for sugar beet and Rhizoctonia. So there are specific regions in the sugar beet which enrich then specific antagonists towards Rhizoctonia, which altogether make these cultivars more tolerant or resistant towards soil-borne pathogen. And a similar we have seen for verticillium in oilseed rape. So verticillium is a normal endophyte also in oilseed rape, but if the abundance is too high, so then the plants get sick and get verticillium wild, and this caused an enormous yield loss. And what we have seen here is that resistant cultivars, they enrich Serratia and Penibacillus. So, and we have also seen that for pumpkin, I have already showed you. So Another very important step was that discovery of the importance of the seed microbiota, which is transferred from one generation to the other. And what we have done here is we studied 2 tomato cultivars, Hilderweiss and Moneymaker, and into soil qualities in loamy soil and sandy soil. And we used here an amplicon sequencing assay in parallel to the cultivation. And for the cultivation, we could check the function or the interaction with the plants. And then we found that it is specifically that the plant beneficial bacteria are transferred from one generation of seeds to the other. But it's not only the seed, it is also the soil, which provides a reservoir for the archaea. We studied that as well. And interestingly, the archaea, they are, although several of them have also plant beneficial interaction, they are much, much more enriched from soil. And our overall conclusion is that there is the association with bacteria is more close. And so especially the plant beneficial bacteria are transmitted by seeds, but there's also an enrichment from soil of bacteria, much more from archaea, and this contributes to the local adaptation, because we have seen huge differences between also the soil quality and the cultivar. So both are influencing this selection capacity. But at the end, it's a good system to have to transmit the best and also to adapt then the next generation to the local conditions. And also here for the seed microbiome, I would like to make a short excursion with you, because we were really interested, because the step was first there were mainly no pathogens, seed-borne pathogens known. Then we discovered the beneficial also in all our crop cultivars. And then we were interested, how does it look like for native plants? And this is here in the surrounding of Graz. We have here these 8 alpine plants, and we want to see which microorganisms are associated to their seeds. And this is a highly diverse ecosystem, which is there for centuries. And due to the fact that these plants grow there together, we expected that they share also a high proportion of their microbiota. It was here, the seeds, they are very small, and we collected a lot also from different sites to make a statistically representative sampling. And we also visualized the bacteria inside of the seed cells, because it's always also important to see that. And so, but we were really surprised by the results. So there were 2 results which we not expected. First is that very little proportion of bacteria and fungi and archaea was shared between the plants. So if you see here these immense diversity associated with the native seeds, these are 12,000 OTUs from bacteria or 4,000 for fungi, and then they share only a few OTUs. So this was really surprising. And so we explained that with the importance for the whole ecosystem, so that it ensures also the diversity of the soil microbiome as a microbiome seed bank, for example. So the other surprise was that we found a lot of potential pathogens inside of these seeds, although all plants were really healthy and we could not see any sick plant. So, and that means also, I think, that diversity matters, and is that these potential pathogens in nature, they have maybe another function, and they are there in low abundance, and we have a lot of man-made resistances. To sum that up is also the seed microbiota especially reflects the core of the plant microbiota. And from our studies, we learned that a healthy seed microbiome is highly diverse, rich, and evenly structured, and it harbors beneficial as well as pathogenic members. And here, again, some pictures from the seed microbiome. You can see them here inside and also surrounding the cotyledon and the root hypocotyl. So, and this is one of our last study on the seed microbiota, that this is really interesting also to find their specific strains or specific species of bacteria which can make the whole cultivar completely resistant, what we have not expected before. So, and we have some ongoing seed projects on the native seed microbiome, because we are interested to see or to learn about the functioning during the germination procedure. And also for oilseed rape, we are doing that. And then we have also a project with a company which then we want to develop the seeds of the next generation. So to sum up, that whole part is, although it was not a systematic study, but altogether we have seen that there we can find already a signature of the Anthropocene in the plant microbiome, and you can, it's visual in the diversity, which is first increasing in intermediate disturbance and then decreasing. The evenness is in parallel. We have a specificity which is reduced. This enhances dysbiosis and also contributes to outbreaks and to more diseases. We have also more R strategists in the plant microbiome, so these fast-growing microorganisms, those which have the potential to mutate even more, and those with antimicrobial resistance or more antimicrobial resistance. And altogether, I think it is essential to start to use the potential of the microbiome to develop a healthy microbiome in form of microbiome management, because I mean, these interlink of the plant microbiome with humans and with our ecosystem, I think it shows also very nicely the importance for One Health or planetary health. So to develop microbiome management options is also important to have new technologies. We have two developed. One is a formulation technology for microbial strains and consortia, which was not available before. And the other one is based on antimicrobial activity these are bacteria-derived compounds. To show you how can such a microbiome management system looks like, here I would like to show you the sugar beet example. So these are 15 years of research and also cooperation with a sugar beet seed producing company. And for them, we developed together with them, we developed 4 products, which are not all commercialized, but this one is commercialized. It's a stress protecting agents for germination, for better germination, and protection against abiotic stress in early life of the sugar beet plants. And then we have also a product which works against pathogen. We developed a monitoring protocol which allows the prediction of healthy sugar beets in the field and also in post-harvest. And we also developed a post-harvest protection cocktail because 40% of the sugar beets are lost after in the post-harvest period. So I would like to show you also that figure because I think it's also interesting. This shows how you can manage the microbiome. And this is completely in parallel for humans, animals, and plants. Because what you can do is you can change the abiotic conditions. So for human, this would be you change your diet. In agriculture, you can change the soil, so that you can change the pH or add something else. So then you can also transplant the microbiota, native microbiota. One of the most successful examples is a fecal microbiota transplantation, which is done since 1,000 years for humans and is very successful. And we have it also for plant microbiome management. For example, if you think on suppressive soil, so and this suppressiveness towards pathogen, you can also bring to another field. Then you can develop pre, and symbiotics for inoculation. So, and also both lines going into the same direction of precision treatments and personalized medicine. So for the management, I would like to show you also the link between plant health, human health, and planetary health. And one of the good examples are apples. And here with the apples, we selected them as a symbol because this was a study which we have done with pupils from school to teach them that microbial diversity is essential for health. So, and I mean, it was also for us new, and we were surprised that there was such a clear message of the management system on the quality of the apple fruit microbiota. So first, each apple contains the same amount of microorganisms. And these are 100 million of bacteria. But if you look on the composition is completely different in organic or in conventional apples. If you look here on the seeds, you can easily see it. And another interesting observation was that most of the microorganisms are sitting in the pulp and in the seeds. So they are not on the surface. So, and if we eat these apples, for sure, some of them are also able to establish in our gut system. So, and therefore, they can contribute to human health as well. We have had it's an ongoing cooperation with South Africa, and we have done a study here, and we analyzed 2 different cultivars of apples, Braeburn and Gala, and we bought them, or we harvested them here fresh in South Africa, and also so Austria imported a lot of apples from South Africa. And what we have seen, and we also compared them with fresh apples from Austria as well, is clear. So, but what we have seen is that although the apples look completely the same, the composition of the microbiome changed from Pseudomonas to Enterobacter. And what is maybe more important is that we have even a storage specific resistome. So there are more resistance genes associated with long-term stored apple. And if you look on the globalization of agriculture, then you can see that a lot of our fruits, vegetables, and agricultural products, they are really globally transported and also stored for a long time in between. And this is really a risk factor then at the end for human health. So, and on the other side, these the post-harvest treatments which we have at the moment, so they are really chemical based, and it is interesting that all the Green Deal and the reduction initiatives, they take care on the field, but on the pre-harvest, but not that much on the post-harvest. And I think here it is also very, very important to develop biological alternatives, and also those considering the microbiome and preventing especially these specific storage resistome. And to learn more about the impact of the food microbiome on the human microbiome and human health, we are here part of a huge European project, and it's called HEDIMED, and it's in frame of the Exposome initiative of the European Union. And we are interested on the impact of the microbiome on the childhood and the early life exposure from different microbiomes to human health. And this is very interesting, and it's very difficult to figure out that, but hopefully we will do. And the advantage is that in that project there are a lot of cohorts, and these are children, and they are monitored each year, and we have a lot of samples from them. And so, and then there are specific groups developing asthma or other allergic diseases. And then you can go back to their early lives, do samples, and look what is the correlation with the microbiome or with other factors of the microbiome. So, and in frame of that project, we are starting to understand much more from the food microbiome, and here's only one result. And you can see that the diversity of apples in Austria is much higher in the private gardens than if you buy them in the supermarket, which are commercially produced. And interestingly, also the apples, so they harbor a lot of human probiotics. So if you eat an apple, so you eat the 100 million of microbes, but you eat also a lot of potential probiotics. And I think this is a very interesting topic of the future, to understand these interconnected microbiomes. And it's not only that for agriculture. So if you look into the native environments, so all the ecosystem, they suffer. If you think on the coral ecosystem, but they are much more. And it seems that the development of probiotics or microbiome management is the only thing what can help our natural ecosystems. So, and this is the end of my talk. So, and yeah, I think I could show you that the global and local microbiome is interlinked, and that we need also the microbiome management to have a healthy future. And so recently we also designed a MOOC. So this is a massive open online course, which is available for free for students, but also for the whole people, for all people, to learn more about the microbiome, and also specific function, and the human microbiome, plant microbiome, and also on planetary health. So thank you very much. This is the end of my talk.
Okay, thank you, Gabriel. Thank you. That was really interesting. So for the participants, now it's time for questions for Gabriel. So if anybody wants to interact directly, can have raise their hand, or either write the question in the chat, and I can ask her directly. And but for me, I have myself a question, because I thought interesting that there is a specific microbiota associated with plants in the Alps that you have found. But I was wondering, because the insects are, especially bees, can be a common vector for some of the plants. So have you looked at the microbiota of the bees to see if there are specific microbes there? So because that will tell you some interesting things about the relations of that bacteria and the plants.
I completely agree. We have not done that, but we have made another interesting observations, and this was the first generation of seed which the plant produce is normally eaten by insects. Mm-hmm. And this I found really interesting. Mm-hmm. And it was really challenging to harvest these seeds, and but because the first generation is eaten, then the second generation is for the plants, and the third generation is for the soil diversity. Right.
No, that's interesting to analyze those insects, to see what is, yeah.
But it would be very interesting to look deeper into that.
Right. So we have a question about of Luis Gonzalez, so maybe he wants to make it directly.
Okay, do you hear me? Okay, first, congratulations, Gabriel, for your nice presentation. I work on the field of post-harvest pathology, so I'm really interested in the development you have presented regarding the apples microbiome and the possible exploitation of the results. So I was wondering, you know, that any product that has to be developed for plant protection has to undergo through the EFSA approval. And as a matter of fact, in the last years, I think there have been only a couple of biocontrol agents being approved candidate. So what is your thought about the possible development of consortia that have been developed through the microbiome studies to be developed as biocontrol consortia, taking into account the European requirements?
Yeah, there is really a huge gap between the scientific results and the authorities and industry. And registration is one of the most important hurdles. And it's also interesting, if you look on probiotics, it is very, very difficult to register them for plants, also cocktails. Yeah. It is much, much easier for humans. So you don't need all these things to do for humans. And for the environment, there's no regulation. And for food, there's also, this is also less regulated. So unfortunately, this is really a problem, and it's very, very difficult to solve because there are a lot of political issues and a lot of economical issues, because also the chemists and the chemical industry has a huge lobby on it. And they are also dominating these authorities, because now they are all working a little bit into that direction. But if you see, there's only Bacillus on the market, and Bacillus is like a chemical, more or less. I mean, this is, yeah.
Okay, thank you very much, Rafael.
Thank you, Luis, for your question. So if there is some other question, you can either write or tell me. Otherwise, I have to say thank you very much again for accepting our invitation, and it was really a pleasure. We have learned a lot of microbiomes in plants, and yeah, I think you should keep working on that because it's a really interesting theme and it has a lot of potential for even for our microbiome. So yeah, thank you very much, Gabriel. There is also other people thanking you and congratulating you, because yeah, I think it was a nice talk.
Yeah, thank you very much.
Thank you, Gabriel. And everybody, see you. Yeah. See you on the next talk. Bye bye, Gabriel. Bye to everybody.